In this video, I want to diagram some of the mechanics of meiosis. And in particular, I want to focus on what divides and when they divide. Remember, in meiosis, there are two chromosome divisions that take place, unlike mitosis, where there's only one division. So let's take a look. We're going to start out here with a cell that has 2n equals 4 chromosomes. And I'm going to show two of the chromosomes as straight lines and two other chromosomes as squiggly lines. So these are the squiggly homologs and those are the straight homologs. So if you will, this is a cell that has its chromosomes in interphase. And you have to imagine that they're all relaxed. And so in the S stage of interphase, they're going to replicate. And so now we have each chromosome made up of two sister chromatids attached in a centromere. The cell now moves from interphase into G2 and then enters prophase. And in prophase 1 of meiosis, the homologs find each other and pair up into what are called tetrads. And they, in this cartoon cell like this, might look something like this. The straight homologs find each other. And pair up. And the squiggly homologs find each other. And they pair up. So that's prophase 1. But you also have to remember in your imagination that in prophase the chromosomes condense quite a bit. And I haven't really shown that step. So here we have prophase 1. And remember, I'm not going to be showing you chiasma and crossing over in this video. I'm focusing on how they divide. So now as the cell moves into metaphase 1, the chromosomes line up on the metaphase plate. So let's say the metaphase plate runs vertically through the cell, and so in that case, the chromosome tetrads line up like so. Alright, so there are our two sets of homologs, the straight homologs and the squiggly homologs, lined up on the metaphase plate in metaphase 1. Now, when this first division happens in anaphase 1, the homologs separate and go to opposite poles. They don't split at the centromere the way they do in mitosis. The homologs simply separate, so it will look something like this in this cartoon version. And I'm going to emphasize the centromeres to show that they're still together. So here we have the two sister homologs, two sister chromatids making up one homolog going over to the left pole, and the two sister chromatids of the other straight homolog going to the opposite pole. And the same happens with the squiggly ones. This squiggly homolog made up of two sister chromatids goes to the left pole, and the other squiggly homolog goes to the right pole. So this is anaphase 1. In telophase 1, then, the chromosomes have moved to the opposite poles, and so over at this pole we have a straight homolog and a squiggly homolog, and a nuclear membrane may develop around it, and then on the other pole we have a straight homolog and a squiggly homolog. So this now is telophase 1. All right, we've done our first division, and the division again is to separate the homologs. All right, let's see. The cell goes through another interphase between the two divisions of meiosis, stays in meiosis, but no replication occurs in this type of interphase. And so now the cell begins its second division. And so let's assume that they're in prophase, and the nuclear membrane has broken down on both poles of the cell, and they're going to line up on the metaphase plate in metaphase 2. And so I'm going to show both sides of the cell. Here we've got the straight homolog and the squiggly homolog of one pole lined up on the metaphase plate. And just for variety, I'll put the squiggly on top here and the straight on the bottom on the other pole. So now we're in metaphase 2. In anaphase 2, the sister chromatids split at the centromere. And so, I think I had the straight ones up here. And squiggly down here. Spindle fibers pulling them away. And the same thing happens at the other pole. I think the squiggly was on the top here. And the straight down here. And so notice that the centromeres have split and sister chromatids are separating. So in the second anaphase, sister chromatids split. This is just like the type of division that happens in mitosis. So in the text I'll say that the second part of meiosis is a lot like mitosis because of the type of division that takes place. So then, by the time we hit telophase 2, and we have the nuclear membranes developing, I'm going to first show the nuclear membranes, and then inside them we have one sister chromatid that's straight, one sister chromatid that's squiggly there, the same thing here, and here. And so we have 1, 2, 3, 4 cells that result from meiosis, and this would be then telophase 2. So the first division separates homologs, and the second division separates sister chromatids.